Hey, it's time for photosynthesis, the last of our chapters that are directly related to metabolism. Anything that's going on inside of a cell that is a chemical reaction is part of metabolism, but this is the last of our metabolic pathways in terms of looking directly at catabolism and anabolism. We all know that photosynthesis is critical to life on Earth, that the organisms that can photosynthesize capture the carbon dioxide in our atmosphere and can convert that into usable energy for those of us who cannot photosynthesize. And this map is showing the distribution of photosynthesis, at least in organisms that use chlorophyll A. Land mass, obviously, wherever there's green plants, but also in the oceans, where there are algae that can carry out photosynthesis. This table shows us the different ways that we can look at metabolism aside from just going through the actual chemical breakdown. So we look at the term trophism, where troph means nourishment or growth. It comes from the Greek word T-R-O-P-H-E, meaning nourishment. And we categorize them based on their energy source and their carbon source. In photosynthesis, we are going to be looking at the photoautotrophs. In each of these names in the first column, the energy source comes first and the carbon source comes second. So photo, the energy source, is light, and auto means self, meaning they can synthesize their own foods from inorganic carbon dioxide. So plants, and algae, some bacteria, and protists can all do photoautotrophic metabolism, regular old photosynthesis. Chemoheterotrophs. Chemo says the energy source is organic carbon, and that organic carbon is also the carbon source. So we get our carbon from the organic molecules that are ingested, and that breakdown is also the energy source. So any organism that can do aerobic cellular respiration is a chemoheterotroph. Photoheterotrophs use light for their energy source, but they cannot synthesize organic matter from carbon dioxide. So even though light is their energy source, they need organic carbon as their carbon source. And it's a very restricted group of organisms that can do this. They're called the purple and green non-sulfur bacteria. So they are prokaryotic. They are purple or green because those are the pigments they use to capture that light energy. And non-sulfur because there are other organisms doing a different type of metabolism that does require sulfur. And lastly, we have the chemoautotrophs. They are using inorganic matter as their energy source. We'll see an example of that later. Things like hydrogen sulfide as an energy source. And since they're autotrophs, their carbon source is carbon dioxide. This is an even more restricted, funky group of organisms, members of the archaea, the ancient ones, that domain that is more closely related to eukarya than the bacteria, but they tend to live in very extreme environments. In our aerobic cellular respiration, we looked at chemoheterotroph metabolism. In this chapter, we are going to be looking at the photoautotroph metabolism. So there's a fern, there are some algae. These are cyanobacteria, so they are prokaryotic. Those are not chloroplasts because it's a prokaryotic cell. They are inclusions of chlorophyll. And in image D, we have the chemoautotrophs. They are using hydrogen sulfide as their energy source and organic matter as carbon dioxide as their carbon source, living at the bottom of the ocean. So that is why the hydrogen sulfide is their energy source. It is so deep in the bottom of the ocean, there is not enough usable light to be a phototroph, to use light as their energy source. This is lit up because there was a camera with a flash on it. There is no light down here. Using deer as an example of chemoheterotrophs, who are primary consumers, they eat only plants, so they rely on the photosynthesizers to get their energy. Looking at the cycle of energy in water, all energy on our planet originates with sunlight. The photosynthesizers take in carbon dioxide from the atmosphere, water through the roots, and can release oxygen as a waste product, as well as the sugars or other organic molecules that we chemoheterotrophs require. Just like we had the very basic carbon, hydrogen, and oxygen formula for cellular respiration, here is the very basic carbon, hydrogen, and oxygen formula for photosynthesis. Taking in carbon dioxide and water in the presence of light, not necessarily sunlight, we can get glucose and oxygen. Now we know, just like with cellular respiration, which said if we combine glucose and oxygen, we get carbon dioxide and water. If we had glucose and we left it on the table in the presence of oxygen, we are not going to appreciably convert that to carbon dioxide and water in any reasonable length of time. There were all of those reactions that were required, and each of those reactions had an activation energy to overcome by enzymes. Likewise, if we had carbon dioxide and water, so some Perrier sitting on the table, and we left it in the sunlight, or we shine a really bright light on it, we are not going to get sugar and oxygen out of that. This is an endergonic reaction. We have to supply energy in because lots of entropy with our reactants, much less entropy with our products. We're creating a more complex molecule. So we have activation energy to overcome, and we actually have a delta G, free energy change, to overcome. So we have to provide the energy from light, and we are also going to have to provide enzymes for this to happen. We don't directly make glucose. We actually make 2 3-carbon molecules that the book alternately calls GA3P, which is glyceraldehyde 3-phosphate. So a 3-carbon sugar with a phosphate on the 3rd carbon, and that molecule is also called G3P for glyceraldehyde 3-phosphate. In eukaryotic cells, photosynthesis happens inside the chloroplasts because the chloroplasts hold the chlorophyll and other photosynthetic pigments. The chloroplast, as we discussed way back in our chapter on eukaryotic cells, does have 3 separate membranes: the smooth outer membrane, the smooth inner membrane, and then a series of interconnected thylakoid membranes. Each little disc is a single thylakoid. If we have a stack of thylakoids, that is 1 granum, and the connections between them allow for water, nutrients, and everything else to pass between the separate granum stacks. The fluid inside that chloroplast is called the stroma. That is analogous to the matrix inside of a mitochondrion, and in turn, the stroma inside of a chloroplast and the matrix inside of the mitochondria are analogous to the cytoplasm of the cell, that aqueous fluid where reactions can take place. So the photosynthetic pigments are inside this third membrane, the thylakoid membrane. There is a space between the inner and outer membranes, but the more important area is going to be the stroma when we look at our photosynthetic reactions. We are going to look at photosynthesis as separate functioning processes, just like we separated cellular respiration into glycolysis and the transition step and the citric acid cycle. But those are not happening completely independent of each other. They're all happening at the same time, all the time, and overlapping, but we have to look at them independently or we can't see where the carbon is going. Likewise, for photosynthesis, we're going to look at the light-dependent reactions and the Calvin cycle, which is also called the light-independent reactions. By their names, the light-dependent reactions do require light energy. The Calvin cycle does require light energy in that it needs the products of the light-dependent reactions, but it happens whether or not light is currently present. That is why it is called the light-independent reactions. I am so old that they used to be called the dark reactions, but they are not called the dark reactions because they will happen when light is present. They are just not directly dependent upon light energy. During the light-dependent reactions, that is when the cell is completely and directly converting light energy into usable cellular chemical energy. And the form of that chemical energy is familiar. ATP and a reduced electron carrier, NADPH. Cellular respiration had NADH. NADPH has a phosphate. So we start with NADP+. During these processes, it gets reduced, it gains a hydrogen, and becomes NADPH. The role of NADP is the same as the role as NAD. It is there to capture those hydrogens that are popped off during the process of oxidation reactions. As chemicals get oxidized, the hydrogens need to go somewhere. They are captured by this coenzyme, by this electron carrier, NADP+. We also input the water. The light reactions are taking care of the water reactant. That light energy is absorbed by the chemical processes in the thylakoids, and the water coming in has its hydrogens removed and it exits the light reactions as oxygens. So the hydrogens that come off of water are captured by NADP+ and exit the system as the reduced form, NADPH. During this process, we are going to be doing electron transport and chemiosmosis in exactly the same way as we did it for cellular respiration. The carriers of the electrons will be different, but their role is exactly the same. When these hydrogens are removed, they're going to donate their electrons through electron transport. That will go through an ATP synthase that will provide the mechanical energy necessary to place this phosphate onto an ADP and make ATP. In the module for cellular respiration, I did put a video of the ATP synthase. It is a really excellent animation that directly shows where are the hydrogen ions going, how are they entering and exiting that ATP synthase, and how does their movement through that ATP synthase provide the mechanical energy necessary for that anabolic endergonic reaction of combining phosphate and ADP to create this chemical potential energy molecule of ATP. The light reactions, the energy required oxidizes water to oxygen, and electron transport is the energy necessary to make ATP. We're also going to reduce our electron carrier, and these energy molecules will enter the Calvin cycle, also called carbon fixation. The carbon isn't broken, but we're going to fix or attach it to an organic molecule. And carbon fixation, when we add it to an organic molecule, we are adding hydrogens. And reduction is the gain of hydrogens. So we are going to reduce carbon dioxide to an organic molecule. Those reactions happen in the stroma, the fluid aqueous portion inside the chloroplast, but outside of the thylakoids. That is where the enzymes for this process are. The supersimplified form of the Calvin cycle is that carbon dioxide enters, and using the energy from ATP and the hydrogens from NADPH, we generate that glyceraldehyde 3-phosphate sugar. And then we have to do it again to get a second 3-carbon sugar, so that we end up with our glucose 6-carbon sugar. Nice little picture of, looks like a Walmart, judging from this symbol, of a bunch of crap food. But all of this food that we eat had to originate with organisms doing photosynthesis to make, well, at least what little organic matter is in any of this food. Photoautotrophs are how we're going to spend the rest of our chapter on photosynthesis. The photo light is the energy. Because they can feed themselves, that means they can take inorganic carbon and fix that or reduce that into organic matter. A super brief electromagnetism mention that light can travel as waves and as particles, and we're going to see both of those in photosynthesis because the pigments in the thylakoids are specific to certain ranges of wavelengths of light. So the length of a wave is the distance between any two crests or any two troughs. And the energy depends on the wavelength. So our sunlight energy or any white light energy is in the visible spectrum. And that is our nice little rainbow. It's shown as the purple to red. If you memorize the colors of the rainbow with the mnemonic Roy G. Biv, stands for red, orange, yellow, green, blue, indigo, and violet. The order here in the spectrum is really violet toward red because of the length of the waves and the amount of energy contained in those waves. As we go from left to right, we go from very short waves to very long waves. So the violet waves are the shortest in the visible spectrum, and the red are the longest in the visible spectrum. Shorter waves have higher energy because the waves have more frequent impact. How many times they hit the object that they're bumping into. And the longer waves have lower energy because a wave will hit you less frequently. Related to our tide reports, if you listen to the marine report, they'll tell you about the swells in the ocean, how tall they are, and how frequent they are. Now, obviously, waves of water are not the same as electromagnetic energy, but the amount of energy contained in those waves is analogous. If you have a 2-foot wave and the period between the crests, the wavelength, is a minute, if a 2-foot wave hits you, you have time to regain your balance before the next 2-foot wave hits you. But if there's 20 seconds between waves, you may not have as much time to recover. You're being hit by waves more often. So short waves, high energy; long waves, low energy. Here's our visible spectrum. It is really, really tiny compared to the full spectrum of electromagnetic energy. If you go beyond, get my finger pointing the right way, beyond the violet, you get into ultraviolet, even shorter than the violet light. And ultraviolet rays, we know, can damage your skin. The way that that happens is because of their short waves, they have high energy, they can actually penetrate our top layer of skin and damage the DNA in our cells and damage the protein extracellular matrix of our cells, sun damage. If they damage the DNA enough, it can cause skin cancer. If we go beyond the red, we get to infrared. The type of red lights in, say, TV remotes and that sort of thing. If we get even shorter than ultraviolet, we get down to X-rays that we know can penetrate all the way through us so that we can see it inside of our bodies, and gamma rays that are even shorter. So these are very damaging types of light. If we go the other way, much longer waves of light, then we reduce the amount of energy and we have things like radio waves, which we're being bombarded with all the time, and we don't feel them or suffer any results from them. So there's our violet to red. We can look at a very cartoony variation in the amount of energy contained in them. Photosynthesis is going to be working more in the green to red areas of wavelengths. Here is looking at chlorophyll A, chlorophyll B, and beta carotene, common photosynthetic pigments. Chlorophyll A and B are identical except for right here. Chlorophyll B has a aldehyde carbonyl on this group, and chlorophyll A does not. That small difference shifts their absorption of light. Chlorophyll A in the solid line, chlorophyll B in the long dashes, so it shifts both peaks. And then beta carotene is a completely different colored pigment and absorbs even differently in the tiny dots. In our forests, especially ones with super tall trees or densely populated, the plants are going to have to adjust. The ones that are the tallest are going to be exposed to the most light, and the ones down below on the forest floor are going to have more filtered light. And they can adjust their levels of chlorophylls, the relative concentrations of which chlorophylls they have, and how much chlorophyll, in order to be able to effectively harvest light. Getting down to the nitty-gritty of what's really happening. This membrane is the thylakoid membrane. And we have complexes called reaction centers that are embedded in the thylakoid membrane. And that is where we harvest the light energy into chemical energy. We have the pigment molecules embedded in the thylakoid membrane. The light comes in, and what happens is that excites the electrons in those pigments and transfers that energy to a primary electron acceptor in the center of our complex, and that is the reaction center. And we have photosystem 2, abbreviated PS2, also called P for photosystem 680, because the maximum absorbance of the pigments in the reaction center is 680 nanometers. That's the wavelength of light. There are other pigments in here that will also absorb light, but the maximum absorbance is at 680 nanometers. And at this photosystem 2 is where the water gets oxidized. We remove the hydrogens from the water. We release that as 2 protons, and the electrons that go in get excited by that energy transfer from light. So the water is the source of the electron that's going to be getting excited by the light. Photosystem 1, PS1, also called P700 because its maximum absorbance is 700 nanometers. And 20 nanometers doesn't sound like a lot, but in terms of wavelength and energy, it does make a difference. These electrons came from the excited electrons from PS2. We harvest light again at this 700 nanometers to once again excite this electron at the reaction center, and then we will pass that on to form more chemical energy. Looking at that on the whole thylakoid. This membrane represents one thylakoid membrane. Here are the photosystems. These blue blobs are the electron transport chain. Different carriers than we had in cellular respiration, but carrying out the exact same function. And we have electron transport happening here between the two photosystems and after this photosystem. We have the electrons originate with water. Light comes in and gets those electrons excited. They pass through electron transport, just like in cellular respiration. As we pass the electrons, we are pumping protons. Here, the protons are ending up in the thylakoid lumen. Nothing else is really happening in here, so it's okay and necessary that the hydrogen ions end up in the thylakoid lumen, because as we increase the hydrogen ion concentration, we decrease the pH. Low pH is a high hydrogen ion concentration. Those hydrogens are eventually going to go through the ATP synthase. This photosystem, where the water enters, is photosystem 2, or P680. This photosystem is photosystem 1, or P700. The reason that photosystem 2 comes before photosystem 1 is that this one was discovered first and they were too stubborn to change the names. P680 is a lower number, so that's how I remember that PS2 comes first. So the electrons are excited in the P680 photosystem, passed through electron transport. That's where we're pumping the hydrogen ions into the thylakoid lumen. The electrons continue to P700, are excited again by a second amount of light, and those electrons go through a very short electron transport to ferredoxin and NAD plus reductase. So it's an enzyme that reduces NAD plus, and that is where we make our NADPH. Since it's outside the thylakoid, but inside the chloroplast, the NADPH gets produced in the stroma. The hydrogen ions that were pumped from the first electron transport into the thylakoid lumen go through an ATP synthase that functions exactly like an ATP synthase in cellular respiration. High concentration of hydrogen ions on this side of the membrane, low out in the stroma, facilitated diffusion through the ATP synthase is the electrochemical gradient that causes the mechanical energy that combines ADP and phosphate into ATP, also in the stroma. Light harvesting happens on the surface of the thylakoid membrane, but the energy molecules, the NADPH and the ATP, are put into the stroma, because the stroma, that aqueous fluid inside the chloroplast, but outside the thylakoid, that is where we're going to carry out the light-independent reactions, the Calvin cycle. Now we see the whole chloroplast, a giant thylakoid, so we can see what's going on. This is the exact same image from our previous slide. Here is our ATP in the stroma. Here is our NADPH in the stroma. And those two energy molecules are necessary to the Calvin cycle. Where carbon dioxide comes in, we have a series of intermediates that keep recycling, just like our intermediates kept recycling in the citric acid cycle. And we're going to need ATP and the NADPH to create the glyceraldehyde 3-phosphates that will exit and eventually be put together to make the sugar. A little more detail on the Calvin cycle. Each turn requires 3 carbon dioxides. That is because we're going to create a 1, 2, 3 carbon molecule. So 3 carbons enter as individual carbon dioxides. They're going to exit as glyceraldehyde 3-phosphate. I apologize for abrupt transitions. I had to sneeze. So here is the Calvin cycle. We have 3 carbon dioxides entering because we're going to exit 1 3-carbon molecule. And 1 3-carbon molecule is the chemical equivalent of a half of a glucose. And that means we'll have to do all of this a second time with an additional 3 carbon dioxides, which works great because the original formula for photosynthesis said that we needed 6 carbon dioxides and 6 waters to come in order to produce 1 glucose. So 3 carbon dioxides come in, we get half a glucose. The second 3 carbon dioxides come in, we get the other half. The 3 stages of the Calvin cycle is 3 molecules of carbon dioxide come in and react with this RUBP, which stands for ribulose bisphosphate. Ribulose is a 5-carbon sugar. It's related to ribose, but it has a different structure. And it's bisphosphate because there are 2 phosphates. So it is a 5-carbon sugar. We have 3 of those. Each of those will combine with 1 carbon dioxide because we have 3 carbon dioxides. The enzyme rubisco does combine that into 3 molecules, each of which have 6 carbons, but those are immediately converted into 6 molecules that each have 3 carbons. Did that make your head hurt? It made my head hurt. I had to ask our botanist years ago. It's like, all right, so if we have 5 carbons and each of them gets 1 carbon, shouldn't we have 3 molecules, each of which has 6 carbons? And that never shows up on any image of the Calvin cycle. And the answer is, it does exist, but it exists so briefly that it is immediately converted to this phosphoglyceraldehyde molecule. That's the best answer I can give you. That is the answer. But if you count the carbons, we have maintained the right number of carbons. This is 3 times 5 is 15. This is 3 times 1 is 3. 15 plus 3 is 18. 3 times, 6 molecules times 3 is 18. Carbon balances. So we have fixed inorganic carbon dioxide onto an organic molecule, and so now we have hydrocarbons. There's oxygens also, but there's hydrogens. We have reduced the carbon dioxide. The next stage requires a lot of energy. 6 ATPs come in, and we're going to be reducing the phosphoglyceraldehyde by taking hydrogens from our NADPH and adding them to this molecule, and we end up with 6 molecules of glyceraldehyde 3-phosphate. So we're not adding any phosphates onto here, but it does require energy to reduce the phosphoglyceraldehyde to glyceraldehyde 3-phosphate. And the hydrogens come from our NADPH. The rest of the cycle is actually dedicated to reproducing our 3 molecules of ribulose bisphosphate. We made 6 glyceraldehyde 3-phosphates. 1 of them exits, so 5 get to continue. And in order to convert these glyceraldehyde 3-phosphates, which each have 3 carbons, into 3 molecules, each of which have 5 carbons, we are reducing the entropy. So we're going to have to add energy for that to happen, and we do take the phosphates, stick them on the end. So we fix the carbon by attaching inorganic carbon dioxide to organic ribulose bisphosphate. We reduce that chemical to the glyceraldehyde 3-phosphate, and then we regenerate our starting molecule so it can capture the next 3 carbon dioxides that come in. This enzyme is ribulose bisphosphate carboxylase oxidase. It is, I believe, the most abundant enzyme on the planet because of all the photosynthesizers that require it. And it's called a carboxylase oxidase because it is sensitive to the relative levels of carbon dioxide or oxygen. If there's lots of carbon dioxide, we're going to do the Calvin cycle, right? Plenty of substrate to bind to the rubisco and do carbon fixation. It's also a signal to the cell that there's plenty of carbon dioxide available that we need to fix. If carbon dioxide levels are very low, then the affinity, the binding, is for the other active site to the oxidase. And there'll be a different process called photorespiration. Plants that live in harsh conditions, very hot, very dry, still do photosynthesis, but there's going to be variations on that. The C4 photosynthesis is more efficient in warm environments, not necessarily super hot environments. It's called C4 because initial carbon fixation creates a 4-carbon sugar. The photosynthesis we just covered here is called C3 photosynthesis because we create, through the Calvin cycle, a 3-carbon sugar. C4 separates the carbon fixation in different cell types, different levels. The mesophyll exterior cells versus further into the plant, the bundle sheath cells. Carbon dioxide enters. We go through the C4 cycle, which uses phosphoenol pyruvate carboxylase to create a 4-carbon compound, and then that releases 1 carbon dioxide into, or 3 carbon dioxides into the Calvin cycle. We recreate a 3-carbon compound that will bind to carbon dioxide and continue this process. So the carbon fixation is separated into 2 different cell types. And that does help to conserve water loss. And we'll show that in another slide. The CAM photosynthesis separates out the light and dependent and light independent reactions by time, meaning night versus day or cool versus hot. So the cacti are going to do CAM photosynthesis. During the night, the stomata are opened. Those are the pores in the plant that allow for water, carbon dioxide, and oxygen to pass through. They're open at night to prevent water loss because it's cool at night. And that allows the carbon dioxide to come in, and we can do the carbon fixation. When it is bright daylight, the stomata are closed so that we can't lose the water through that, but we can carry out the other photosynthetic reactions. So we can't harvest light in the dark, but we can do carbon fixation. We can't do as much accumulation of carbon dioxide during the day, but we can harvest the light. So if we compare these, and I apologize, this looked better when I put it in to my slide, and then when it's projected, it's kind of blurred. I'll find the link to this to put in the module. But in C3 photosynthesis, it doesn't really matter if it's day or light. The stomata can be open to get the carbon dioxide. And the advantages is that it's very energy efficient. Disadvantages that you can have some water loss due to evaporation. C4, we separate in which cell types the carbon fixation and the Calvin cycle take place. And the advantage is we do minimize water loss, but it does require more energy to do 2 separate cycles. And in the CAM photosynthesis, We reduce the water loss, but it requires more energy and it does slow the growth down. And so there are examples. Typical plants do the C3. Plants like corn that grow in warm environments but not the desert. There's plenty of water. And then the cacti that they're hot and dry do the CAM photosynthesis. It's just showing our relationship between consumers and the producers. This creature does aerobic respiration, which generates carbon dioxide and water. This organism takes in that carbon dioxide and water, produces the oxygen that this animal breathes in, and the carbohydrates that the animal also needs to eat. I will have a summary of this, just like I will do a summary for the aerobic respiration. I did post the aerobic respiration summary and some metabolism worksheets in the modules so that you can do some review for that, and I will post the key to that so that you can compare your answers.